This part is a little bit complicated because we have several types of oligo and trace elements. In fact, all the elements that you can find in the table of Mendeleev are actually physically present in the human body, or could technically be determined in the human body. And several of these components do not have a direct relationship with each other. So it's very difficult to come up with a common ground or a common characteristic for the elements. The most important thing we can say is that most of these elements are present in the human body in a concentration lower than 50 milligrams per kilograms. And so we're really talking about very small amounts, some of them even only micrograms. And these compounds have something to do with the human body. It could be two ways. The first one could be that they are there because they are essential. So they are essential for performing specific reactions. The second option is that they are there because they have a contamination, and that they are just coming into the human body because you are exposed to it, and in principle, they should not be there. If we look to the analytical methods that we have available today, then we have to say that we can analyze very peculiar and very strange elements in the human body. I'm thinking about lead, cadmium, arsenicum, or even boron. So these are all compounds that are there. Probably they're not there for health, but they could physically be there because of some form of contamination. So in order to know whether it is contamination or not, you could look at the distribution of the element in an organ. And take a liver, for example. You could measure the liver at several locations in the organ. If you find the element back in all cases in a quite constant amount, then probably it leads to a form of essentiality. While on the contrary, if you can find it in very strategical locations in organs or in cells, in those conditions, it could probably be a contamination. And for example, the lysosomes are very small particles that you can find in the inter part of the cell, so in the intramolecular region, intracellular region. And it could be that these lysosomes pick up these trace elements and isolate them from the rest of the cell so that they cannot be toxic towards, for example, the cell nucleus, endoplasmatic reticulum, mitochondria, or whatever. Another general characteristic is the fact that some elements like cobalt have no requirements for humans. So if you would say what is the requirement for cobalt, then we have to say we don't know. But we do have a recommendation for vitamin B12. And cobalt is an atom that is located centrally in the vitamin B12 molecule. So for humans, we have a B12 recommendation, but not a cobalt recommendation. And the reason is because humans can only absorb B12 directly coming from the food. So we cannot produce vitamin B12 starting from cobalt. So that's why we don't need a recommendation for that. In ruminants, like cows, for example, there they have a recommendation for cobalt and not for vitamin B12. And the reason is that a ruminant has a number of bacteria in the first part of the GI tract, and the cobalt which comes in can actually be converted by the microorganisms into a vitamin B12, which can then, in the small intestine of the animal, be absorbed to be used in the animal as such. So for animals, ruminant animals, there's a recommendation for cobalt, but not for vitamin B12. In humans and all single stomach species, like for example, pigs, there's no recommendation for cobalt, but there's a recommendation for vitamin B12. We can synthesize B12 in the colon, but we're not sure that the amount absorbed is equivalent to the amount needed in daily life. And then we have a final element that is linked to trace elements, and that has to do with concentration dependence. So it seems that if you are in low concentrations, we are in a zone of deficiency. And it actually means that if you give more of the trace element, then the activity which is linked to the trace element can increase. For example, an enzyme, which is fully dependent on the trace element, can increase in activity when the dosing goes up. So when you consume more of the elements. If you see that concentration dependence as a function of activity, then you are in the zone of deficiency. You can increase the dose further until you reach the so-called optimal function, which is a zone where normally the activity will stay more or less constant. You are in an optimal situation. If you continue even further with higher doses than optimal, you get into the pharmacological dose, which means that the activity will further increase compared to the optimal function. But in those situations, you can go quite fast into the so-called toxic zone, which means that the amount you take in is too high for normal functionality of the human body. So we always aim to be in the area of optimal versus pharmacologic, with a slight preference for optimal, because pharmacological doses might relate into toxic areas. And we can also say that the difference between deficiency zones and toxic zones are not always very large. And sometimes the deficiencies are very small, like for fluor and selenium. We know that fluor is in some individuals in our countries here deficient, which means that these people do not have enough by just cleaning the teeth twice a day and getting in some toothpastes that contain fluor. For those people, it seems that we need extra fluor, but adding fluor to drinking water, for example, is no option, because there may be several other people who have far enough fluor intake for the moment. If they would also start drinking this fluoridated water, probably they reach the toxic zone, which we don't want. So for these reasons, if the area between deficiency and toxicity is very small, you have to be very careful of introducing the element on a large scale within the population. A similar remark can be said for selenium. If we look to the contents in the human body, then you can see that the amounts are really not high. And for iron, it can go up to 3 to 5 grams for a total body, which corresponds to about 30 to 50 milligrams per kilogram. While in selenium, for example, or iodine, the values are smaller than 20 milligrams for a whole human body. And a human body is about 60, 70 kilograms. So it means that the amounts there are really trace. And especially with iodine, for example, all the iodine or practically all the iodine is concentrated in the thyroid gland, and less than 1% is circulating in the human body. Why are trace elements so relevant in human nutrition? Well, the first problem that we face is that it's very difficult to define essentiality and deficiency. So we cannot just straightforward analyze a trace element status in humans. We have some parameters, we have some markers, but as a whole, in many cases, it's still a quite difficult measurement to do. For example, in the case of iron, we know that the chemical form and the interaction between nutrients will finally determine the absorption and utilization. Iron can either be absorbed in the ferro form, which is actually the iron 2 form, and this is the best form to absorb it. But you also have iron 3, which is the oxidized form, and this form is not very well soluble in the intestines and will not contribute to the recommendation. While heme iron, which is, for example, present in meat, is very well protected against oxidation, so will normally always stay in the ferro form, and is quite easy to be absorbed. And so the form of the element will determine the degree of absorption. And this makes it difficult for us to really come up with a recommendation, and also to come up with a strategy to see when the element will be deficient or not, since it has different forms. We also know that it has interactions with other nutrients. For vitamin C, for example, is definitely an element that can interact with iron. It can bring iron 3 back into iron 2, so it can reduce it in such a way that the iron 2 can more easily be absorbed by humans. We also see the excretion, which is dependent from circumstances. The requirements is dependent on the physiology. So there's a difference between males and females, for example. And there's a very difficult measurement. The best way to measure it is probably the liver. But the liver is an invasive technique, so you need to take samples from the liver in humans in real life. It's not easy to do that, and probably it will not happen in this way. We usually take blood samples, analyze the serum concentrations, and use them as a measure for the trace element status in the body. Or we just measure enzyme activity with some enzymes that are directly linked to the trace element status. And the most general way of working is, for example, using clinical symptoms like anemia. But we have to be careful because anemia as such is not the only parameter that is linked to, for example, iron. Anemia can be linked to iron, but it could also be a copper deficiency. And so if you see that a person is anemic, you're not allowed to directly say that this is caused by iron deficiency. There could be other elements involved, in this case, copper deficiency. I will come back to the mechanism in a minute. So these are a few general aspects that I really wanted to share with you because it's such a heterogeneous group with so different elements that it's not always easy to find a common ground to discuss all these elements. We do know that one of the common grounds is the difficult form of utilization. And in order to explain that, I give you the model of homeokinesis. So it's a model that actually explains that the most important regulation step for trace elements is the regulated absorption. So we know at the level of the small intestine that the amount of trace elements that can be absorbed there is extremely limited. One of the arguments to support that idea is that once the trace element passes the absorption stage and reaches the blood, it is very difficult to actually control it further. For example, it's very difficult to excrete it from the human body. If you take iron, for example, it could come in from the intestinal tract, go into the blood, but you will not secrete iron through the urine. So if you have too much iron in, you cannot get rid of it by just urinary samples. It doesn't work like that. So normally, if it's in the blood, there is an equilibrium between, on the one hand, the target organ, and on the other hand, the storage organ. And the storage is usually the liver. The target organs can be manifold, like, for example, the muscles. For example, also the spleen, where you have actually the production of the red blood cells. And measuring or controlling absorption controls the overall status of the trace element in the human body. So you can imagine that when the regulated absorption is damaged, for example, a genetic defect or something like that, that the trace element can freely flow into humans, be physically there in high levels, be transported to all types of organs, and cannot be controlled because you cannot excrete it. So that's why you need to check very careful that the absorption level is very highly regulated. And in humans, normally healthy humans, this is the case. So it is so heavily controlled that the amount you pick up and take in is actually far lower than what we really need. And so we create a problem ourselves in terms of deficiency because the regulating absorption is extremely strong in the human body. I will give you examples of this absorption also in a minute. If we look to the two final major common grounds for trace elements, then the first one is definitely amplification. So amplification means that a very small turnover of the element itself will give a very high metabolic turnover of the metabolism. And I have two examples to illustrate that. If you take cobalt, the turnover of cobalt is 10 to the power minus 10 grams per day. So it's a very small change in cobalt that we see here. But cobalt is a cofactor for vitamin B12. And B12 is a cofactor for various enzymes, like methyltransferase, that plays a role in the synthesis of thymidine and DNA. And DNA is important for transcription, and transcription is important for protein synthesis. And the amount of proteins synthesized can vary between 1 and 50 grams per day, which is a huge difference. And so a very small change in cobalt could lead to a large change in protein synthesis. And this is the reason why the body will keep cobalt concentrations as constant as possible, because slight changes have a very high impact at metabolism level. And the same thing can be said about chromium, turnover 10 to the power minus 9 grams per day, important for glucose tolerance, insulin production, and as such, energy metabolism. So if you change chromium to small amounts, it could give significant differences in terms of energy metabolism. Again, that's why we have to keep the chromium levels as constant as possible. And most of the trace elements have this feature, and so they all amplify when there are small differences in concentration. And the last element is the spectrum of metabolic activities. We know that some elements have a low, limited number of activities, like iodine, for example, while other ones are quite diverse. Zinc has more than 70 different functions in the human body, and it will be very difficult for that type of element to exactly determine the zinc status, because it all will depend on the biomarker that you use to measure zinc levels in humans. A very important phenomenon that takes place in humans is the so-called radical formation. And this is very relevant also in the context of trace elements. So I tried to summarize all these elements here together on the slide, where you can actually see the first step, which is oxygen. So oxygen can be converted into water by electron transfer, and this is done by the electron transfer chain that you all know and that is physically present in the mitochondria, and that is responsible for the synthesis of ATP in the cell. So normally this pathway is useful for us because you can actually, in this case, generate ATP that could be used for general metabolism. Some of the complexes that are involved in the transport chain could also lead to leakage of radicals. Especially complex number 2, for example, is very leaky. You get a lot of radical formation there, which can end up in the cell and which can damage the cell. So in that respect, that could be not so beneficial. We also know that these radicals are produced by an extra series of reactions, starting from oxygen. The oxygen can be converted into so-called superoxide anion, and this can be done by UV radiation, by high intakes of energy, as well as by local enzyme activities. Here you can see the superoxide anion, O2 and then dot and a dash. To explain this in a chemical form, you could say that the molecule of oxygen has different electrons, and one electron will go to a higher level within the molecule. And if it does that, it will retain a higher energy content, which makes the molecule reactive, and then it will respond further by interaction with other molecules, producing, for example, hydrogen peroxide, which is a very heavy reactive molecule because it can destabilize into so-called hydroxyl radicals, which can attack proteins, lipids, as well as DNA, so which is not useful for humans in high quantities. The pathway starting from superoxide anions going to the hydroxyl radicals requires trace elements. So the first step is superoxide dismutase. This one is dependent on zinc, manganese, and copper. And the second step from hydrogen peroxide into hydroxyl radicals is a so-called Fenton reaction, whereby iron 2 is oxidized to iron 3. And in order to bring it back to iron 2, we need copper that can, in this case, be oxidized from copper 1 to copper 2. So you can already see by the red arrows that radical formation can be stimulated by trace elements. And it could lead to unuseful or negative side reactions in the human body. Luckily, we have some options to control it. And these are the blue or green areas. So vitamin C and vitamin E, respectively in the water phase and the fat phase, could neutralize the hydroxyl radicals so that they are less active and will not destroy proteins, lipids, or DNA. The second option is hydrogen peroxide that can be converted into water. The enzyme is called glutathione peroxidase, and it's dependent from selenium. I think that last week we were discussing about glutathione, which contains sulfur, if you remember. This glutathione can actually bind to selenium and create an enzyme, the glutathione peroxidase, which completely neutralizes hydrogen peroxide and which could be useful to protect the human body. However, if we go on with another reaction, which is called the catalase, then you can see that also in this case, hydrogen peroxide can be converted into oxygen and water. And there you see already the difficult role for iron in metabolism, because iron could be negative, producing radicals, but could also be positive, inactivating radicals. So for iron, it's always a delicate balance between negative effects and positive effects, and you never know for sure what is now the optimal level for iron. While in the case of selenium, you see that we are mainly looking at detoxification. So this element could have a positive, useful role to play in relation to radical formation. And radicals as such are important because they can lead to disease processes in humans that could be very variable, like, for example, cardiovascular disease or some forms of cancer. So I hope with this you know the interplay between trace elements, that you know the delicate balance between risk and benefits, and that in the case of iron, we have to be a little bit careful because the balance can go in two ways, either positive, but as well, perfect opportunity to also go negative by stimulating the Fenton reactions. And this brings us then to the first element that we need to discuss, which is iron. It's definitely the most important one because on average, 40% of people could be deficient in iron, which means 4 out of 10. And if we take our classroom and we look at students, if you would take 10 students, 4 of these do not have an optimal iron status, which is quite significant. And if you look here to the data from World Health Organization, these data were actually published in 2005. So we're talking about, almost 20 years ago. And there you can see very clearly that the tropical belt in the center of the Earth is highly at risk of anemia. And the more darker the color you see, the higher the potential prevalence of anemia in the world, and of course, in this case, iron deficiency anemia. Today, the picture is not that much variable. I looked at data from Belgium from the last years, and there we see that the incidences for specific target groups like pregnant women, young children, could still be in the area of 35 to 40%. So we do not seem to be capable of solving the problem as a function of time. And you will understand if we go through the mechanism why iron is so difficult to solve today. It has some major problems to solve. If we look to the incidences, you see that in tropical zones, Africa, for example, Latin America, a large part, some parts of Southeast Asia, there you can see high incidences of iron deficiency. We have to be careful because in some areas there we have malaria, so malaria endemic regions, and it seems that malaria can also contribute to anemia in humans. So if you take anemia as a basic screening method to look at people that are deficient in iron, you have to be careful because at the same time there could be a copper deficiency, which has nothing to do with iron deficiency. And secondly, there could also be malaria incidence, which will also interfere with the test that you are performing. So let's look a little bit at the metabolism. So if you look to iron, then you see that the intake is about 12 to 15 milligrams. The excretion is quite similar. 12 to 14 milligrams of iron leaves the body every day. So the amount that you absorb is extremely small. It's probably less than 1% of what you really physically take in. What is taken in the form of iron 2. So it's the ferro form, not the oxidized form. It should be the reduced form. In order to bring it in the reduced form and to keep it there, you can use copper. So copper is actually the universal counter element for iron, and it can see that iron stays in the right chemical form for normal metabolism. The next stage after the absorption is going to the plasma pool. In total, we have about 3 to 4 milligrams of iron circulating in the blood, and this is done in the form of transferrin, which is a transporting molecule that can bind to iron. And important is that the iron can only be bound in an iron 3 plus form, so the oxidized form. So you absorb it in the form of iron 2, you transport it in the form of iron 3. And once it is transported, it can go, for example, to the liver, where it can be stored. It will also be stored in the iron 3 form. So in the liver, there's no need to do any modifications. However, if you go to the bone marrow, so the location where the red blood cells are produced and where iron is incorporated into heme, there the iron again has to be reduced from iron 3 to iron 2. And this is again a function which is catalyzed by copper. So if you look to copper, it can actually transfer iron 3 into iron 2, and iron 3 and iron 2, in this case for the muscles, as well as for the bone marrow. So a copper deficiency, as you can see here, leads to disturbances in absorption, disturbances in transformation into red blood cells, as well as transformation into myoglobin, which is the protein that is stored within the muscles. So remember, copper and iron can give the same symptoms of anemia, but fundamentally the two are collaborating, but are not the same in functionality. The way to reduce iron is not easy. You can actually have in females the menstruation, generally speaking 28 milligrams of iron each period, while on the other side there are small losses, but it's really not big, in the gallbladder, in the urine, as well as in the sweat. And so you can understand that for the majority of people, it's very difficult to get rid of an excess of iron. Just imagine that your absorption capacity is blocked. For example, there's a genetic defect, so that iron freely flows from the intestines into the blood. In those cases, it becomes dangerous because you have very high levels of iron in the blood, and we can't get rid of it. It will just accumulate and just circulate to all the organs. So the only way to treat it is by just taking regular blood from that patient. And so they have to go into hospital on a regular basis. Blood is removed from the body in order to remove iron from the body. And so you see already drastic applications to go back to a normal status. So this is the situation. Then the question is, what can we do about it? So what do we know about these pathways more in detail so that potentially we can still improve iron status? The first option you have is working in the luminal phase on solubility. And we know that iron in the 2 form is well soluble. Iron on the 3 form is not soluble. So you could try to change the pH of the slurry of the material that is present in the GI tract. For example, iron 2 is soluble, so you could reduce it. And so bring it back from Fe, which is iron 3, to ferro, which is iron 2. You can do that by, for example, using vitamin C, because this one is an oxidation reduction agent. So in your strategy in humans, you can add foods that are quite rich in vitamin C, and this could contribute to bringing iron in the iron 2 form, which is the best way to absorb it. The second option is working with heme, which is present in red meat, for example, and this heme nucleus protects the iron in the iron 2 form, so that also that one will be more easily absorbed in the small intestine. The heme nucleus as such will normally not fully be absorbed, so that one can go to the colon, can be converted by the bacteria, and in high quantities could potentially also be harmful for the colon wall. I think in chapter 3, when we were talking about fermentation in the large bowel, we already explained a little bit how that could contribute to colon cancer. We also have cysteine, which is sulfur, and we have organic acids to reduce the pH that can all help in increasing the solubility and increasing the absorption of iron. If we go to plant-based foods, then we know that the iron bioavailability is low. And this could be due to the presence of phytate, which contains phosphorus, and phosphorus makes a complex with iron and reduces the bioavailability of iron in the GI tract. The same thing for polyphenols and the same thing for fiber. And for example, iron could be bound by fiber. Fiber has a low retention time, so it goes very fast to the fecal material. And in this way, it can drain iron away before it is actually absorbed in humans. So for plant-based material, if you want to increase the iron status, you probably have to do something on the phytate levels, the polyphenol levels, and the fiber levels, and probably by reducing them first by processing so that the iron status and the iron availability increases. Animal feeds have, or animal foods have about 40% of heme, while vegetable foods have 100% non-heme. And so generally speaking, animal foods have a higher source of iron bioavailability, while in plant-based foods, these types of foods have a low bioavailability for iron. Once the iron gets into the small intestine and in the wall of the small intestine, we have the so-called mucosal block theory, which I will explain on the next slide. It means that only after saturation of ferritin, there is the possibility that iron will be transported intracellularly. If ferritin is not saturated, then iron will not pass to the blood. It will always be bound to ferritin until the ferritin is completely blocked and saturated. And then, depending on the demand from the body, slowly but surely extra iron can be released. The transport from iron to the different organs is then done by transferrin. So let's have a look on the mechanism. So it looks a little bit complicated. The article is online on Euphora in case you want to see the original one. I just try to explain to you how the mucosal block works. And it's quite important because this is actually the key element that finds that iron in the human body and absorption in the human body is extremely limited. So let's start on the top, and what we have here are mice assays, whereby we are working with wild-type mouse. And so there is nothing changed. The mouse are just the same as they would be in a normal situation. On the left side, the situation for low body demand, so there's enough iron. On the right side, the situation with a high body demand when there's a lot of need of iron in the body. So what we see is that there are elements, proteins, which we call iron regulating proteins. They are physically located in the cell of the epithelial wall. We have a transporter, which is on the north side, apical side. There's a transporter on the basal side. Here is the bloodstream on the bottom, and on the top you have the gut lumen. So when iron comes in, and we don't need any iron, so there's no demand, then you can see that the transporters are not that well expressed. And so we're only one transporter here, and there's only one transporter on the other side that links to the blood. So not high expression of transporters. And at the same time, we also see that there are iron regulating proteins, which we call the IRPs, that actually lead to more synthesis of ferritin. And ferritin is a storage protein. So all the iron that can come in, and they don't have a lot of chance because there's only one transport channel, so they can just come in by one transporter. There's so much ferritin there that all the iron will be bound to ferritin. And so it will be very difficult for iron to further migrate down the cell and be actually taken up by the blood, especially because there's a second transporter that needs to be passed before you are physically in the blood to be circulated. And at low body demand, we also see that the body in the liver will produce a hormone which is called hepcidin. And hepcidin will block the second receptor or transporter so that you have no supply of iron from the epithelial cell to the bloodstream. So you can imagine we have a double system. So the first transporter, then the ferritin area, and then finally the second transporter. And this is all so tight that iron in practical conditions cannot easily penetrate down into the blood. That's why we call it mucosal block. On the other side, on the right side, there is a high body iron demand. In this case, the proteins, which are called iron regulating proteins, are activated. You have much more synthesis of transporters, both apical as well as basal, and you don't have any production of the ferritin. So in this case, the iron comes in, is transported intracellularly, goes to the other side, and on the basal compartment, it is just released into the blood. And so in this case, it's more easier to absorb iron. So these iron regulating proteins, these IRPs, play a very important role. And in the bottom experiment, they used the same rats. And now they have actually made a knockout of iron regulating protein. So they just destroyed the synthesis of IRP just to see what the result will be on the iron intake. And in this case, we see that there is an uncontrolled synthesis of ferritin and an uncontrolled absorption of iron. So indeed, iron regulating proteins play a key role in the absorption of iron in humans. And deficiencies there will also lead to disturbances in iron uptake and metabolism. So what can we do to improve iron status? And so which options do we have in order to make some improvements and corrections? It's not easy, and the science is already working for more than 50 years in that area, but still today we don't have a clear-cut solution on what to do. The first option is dietary variation. So you could, for example, stimulate production of meat and animal-based products in developing countries. It's a risk because probably we will force people to have higher intakes comparable to what we have in Western Europe and in the United States. But as you already know from the studies there, is not always a beneficial situation. And so it is very delicate. We might increase it a little bit, but hopefully not too much, because then you might end up with the negative characteristics that we already know in the Western world. Second option is supplementation. So if we look, for example, to malaria endemic regions, like in Africa, we know that tablets are distributed for iron. And we also know that in malaria endemic regions, it seems that giving tablets of iron could also lead to higher incidences of malaria in the same area. So giving loads of iron is definitely not useful when we're talking about societies that have malaria. And I already told you, malaria is also influencing anemia. So anemia could also be caused by malaria and not necessarily by iron or copper deficiency. So that's the second option, supplementation. And the third option is fortification or biofortification. Biofortification means genetic engineering. So you take a plant-based food, you genetically engineer the genetic material in such a way that the plant will produce more iron and will produce less anti-nutritional factors, for example, phytates or polyphenols or other compounds. The word fortification without bio is actually interfering during the production process. So you could take a flour, you can add a certain amount of iron to the flour, mix it, and then use the flour for the production of bread. The big problem there is that you need to homogenize the material. And we know that when interventions are done in humans with trace elements, the person who is the most important for coordinating this intervention is the person who is actually milling the product and bringing the trace elements inside the product. Because if a mistake happens there, then some people will have a very high toxic intake of the component, while other people will have almost nothing. And so you need to be sure that the material is very homogeneously distributed inside the flour. Popular countries that are actually fortifying flours today with trace elements, especially iron, are mostly countries around the Mediterranean and at the African side. For example, Morocco, Libya, Algeria, Egypt. These are countries that are normally have a mandatory fortification at governmental level inside plant-based products, especially for iron. So I hope you see there's not an ideal solution. We try to get solutions. We have several options, but we never have an ideal case. If we look a little bit to iron fortification, biofortification, you know, genetic engineering in Europe is not very popular. Consumers do not really want that at the moment. But in several other areas of the world, North America, Africa, for example, Southeast Asia, there we find several studies that have already been performed by using biofortified crops. And there's one study, the study of Finkelstein, which was published about 5-6 years ago. The article is on Euphora if you want to see it. There, 3 human intervention studies done in the South have been combined to evaluate the intake of biofortified crops on iron status in humans. And the indicators which were used were serum ferritin, which is a measure for the storage, total body iron, which can be derived by using ferritin and transferrin, and finally hemoglobin, which is a functional parameter and which transports oxygen through the blood. We have 2 types of individuals, all individuals or iron deficient individuals. And if you look to the last column, there you can see the p-values, then you will see that in many cases, not in all, but in many cases, you can see a significant improvement in iron status in humans, either measured by serum ferritin, which is a measure for the amount which is stored, the total amount of body iron, which is also significantly improved, and we can also see that the hemoglobin levels are also significantly improved, especially in the iron deficient people. If you bring all the people together, then the effect on the iron deficiency people is neutralized. But for the iron deficient people, giving biofortified crops definitely and significantly increase the hemoglobin levels in that patient or person. So technically, it is possible to change iron status with these products, provided that legislation in the community, in the country, allows that it is used for human consumption.